So let's go through and explain everything you need to know about Module 2.1, the digestive system. Make sure to give the video a like, make sure to click the subscribe button and the bell as well so you get notified of any other module explained videos so that you can smash your GCSEs. So we're going to start by thinking about our levels of organisation. Our smallest level of organisation is an individual cell. We could go smaller than a cell, we could talk about our organelles, but cells are their own independent unit of an organism. And a collection of similar cells that perform a specific function is our tissue. A collection of different tissues would then come together to form an organ. So the example we have here of an organ is the heart. The tissues, we have muscle tissue, we have nerve tissue within the heart as well. And then a collection of organs come together to form an organ system. So for example, the heart there makes up part of the cardiovascular system, and that cardiovascular system consists of different organs. And then a collection of organ systems, well, that's going to form an organism. So another example of an organ system could be the digestive system, and that's what we're going to break down in detail. So our digestive system, let's go through our different organs. We're going to start up at the top by the mouth, and specifically in the mouth, we have the salivary glands. Now the salivary glands are going to produce saliva, and within the saliva is going to be the enzyme amylase. We're going to talk more about enzymes in a minute. The mouth also has a role in mechanical digestion, where it breaks down the food particles into smaller pieces, which increases the surface area so the enzymes can work faster. Once the food has passed through the mouth, it is then going to go into the esophagus. Don't worry about the spelling of esophagus. As long as you spell it phonetically and it's clearly the same word, you'll still get the marks. Now, the esophagus passes the food down towards the stomach. The stomach has a couple of adaptations. The first one is that the stomach produces a few enzymes called proteases that help in the digestion of proteins. The stomach also produces hydrochloric acid. Now, the big mistake that people make is they think that the hydrochloric acid in the stomach is there to break down the food. That is wrong. The hydrochloric acid is there to kill any pathogens, perhaps, and that links us to our immune response and defenses against pathogens. And it is also there to provide the optimum pH for those protease enzymes. The stomach also helps to churn the food, which is another form of mechanical digestion, helping to break the food particles down and increase the surface area. After the stomach, the food passes into the small intestine. Now, the small intestine is where the majority of the absorption takes place. Now, there's two different things going on in the digestive system. We have digestion, which is the breakdown of large complex molecules into smaller soluble molecules. So, for example, substances like carbohydrates are too big to be absorbed. They are also insoluble, which means they cannot be dissolved. And so they need to be broken down into smaller subsections or monomers, we could say. And for carbohydrates, that could be things like glucose. Now, those glucose molecules, they are smaller and they are able to be absorbed. And that's the role of the digestive system. In the small intestine, we have the absorption. We do also have digestion taking place in the small intestine, and that is where other enzymes such as amylase and lipases are going to be produced. After the small intestine, we have the large intestine. And the large intestine is where the majority of the water absorption takes place, and that is then going to form the feces, which are going to be stored in the rectum before being egested through the anus. Now, there's some other parts to the digestive system which help in the function of the digestive system. The first part here is the pancreas. And within the digestive system, the pancreas has the role of producing enzymes. The pancreas also has a role within the regulation of blood glucose levels, but we'll be looking at that later. The other part here is our liver. And the liver is connected to the gallbladder. The liver is going to be producing the bile, and the gallbladder will store the bile before it is then kind of secreted into the small intestine. So we have our different parts of the digestive system there. Let's move on to looking at our enzymes. Enzymes are super important in the functioning of the digestive system. In fact, enzymes are super important in the functioning of all cells and all organisms. Enzymes can be described as biological catalysts. What that means is that they are biological molecules. Enzymes are made of protein, and their role is to speed up the rate of chemical reactions. Now, just like a catalyst in chemistry, they do that by lowering the activation energy. So, how do enzymes work? How do they speed up these reactions? Well, we can describe the model of enzyme action using the lock and key mechanism. The first thing we have is our substrate. The substrate is the substance that the enzyme is going to be breaking down. So this could be a carbohydrate, for example, like starch. And then we have our enzyme. And there is a specific region of that enzyme called the active site. Now, the active site of the enzyme, we would say, is complementary to the shape of the substrate. They're not exactly the same, but they match each other, a little bit like jigsaw pieces. And this active site is specific to only one substrate. So, for example, our enzyme amylase will have an active site that is complementary to starch. It won't have an active site that's complementary to anything else. Amylase cannot break down other substances. Once the substrate binds to the enzyme, this is forming our enzyme substrate complex. The enzyme substrate complex then carries out the reaction, and for example, our starch is broken down into our glucose and the products are released from the enzyme. And importantly, the enzyme is unchanged in this reaction. Just like how a catalyst in chemistry is not used up in the reaction, the enzyme is not used up either, and so goes on to carry out the reaction again and goes on to aid in the digestion or the other metabolic reactions happening within the body or within the cell. So there are 3 examples of digestive enzymes we need to know about. Our first example is the enzyme amylase. Amylase is an example of a carbohydrase. There's different types of carbohydrases because there are different carbohydrates and they would all have different active sites that would be complementary to different substrates. Amylase is specific to the substrate starch. We can see starch is our big complex molecule there, and we're going to break down that starch into the individual monomers of glucose. Our next example of an enzyme is a protease. Now, proteases specifically break down proteins, and they are going to break those proteins into amino acids. We can see we've got different colors here for our amino acids. That's because there are 20 different amino acids in the natural world. And the proteins are made up of different combinations and sequences of these amino acids. But the proteases are going to break the proteins down into these individual monomers. Our next enzyme is a lipase. And the lipases are going to break down the lipids. Lipids are our posh technical name for fats and oils. Now, the lipids are made up of a few constituent parts. Lipids are made up of these pink parts, which are the glycerol. And as we can see here, we also have fatty acids. And these lipids have 3 fatty acids. We also need to know where each of these enzymes is produced. Now, amylase is produced in 3 different areas. We mentioned earlier about the salivary glands. So the amylase is produced in the salivary glands, the pancreas. The pancreas is one of our glands that produces lots of different enzymes, and the small intestines. Now, proteases are produced in 3 different places. They are produced in the stomach, in the pancreas again, and in the small intestine. And lipases are produced in only 2 different areas, from the pancreas and again in the small intestine. As well as knowing our examples of different enzymes, we also need to understand how the rate of those enzymes are affected. So we have three main factors that affect the rate of these enzyme-controlled reactions, or affect the speed of these reactions. Our first one is the availability of the substrate, or we could describe that as our substrate concentration. If there is more substrate, the enzyme is going to be able to work faster. The reaction progresses faster. It's just like in chemistry. If there is a greater amount of the reactant, the reaction happens faster. And we can see that in our graph here, where if we have increasing substrate concentration on my x-axis and the rate of the enzyme on the y-axis, we can see this increase over time. Now, eventually we are going to reach a plateau. And the reason that it plateaus there is because all of the enzymes are busy working at their maximum rate. There's no more enzymes available. So this is when we could say that the enzymes are acting as a limiting factor. Our next factor affecting the rate of enzyme-controlled reactions is temperature. Now, temperature affects all things in biology. Temperature is a measure of the kinetic energy of the particles. If they have greater kinetic energy, they are going to be moving faster. And that means that the rate is going to increase. There's going to be more collisions between the substrate and the enzyme, and so more enzyme substrate complexes will form, and therefore we will get more product being produced. But temperature has an optimum where the rate is going to be at its maximum. And if the temperature exceeds that optimum, well, the enzyme is going to be affected. The enzyme is going to be denatured. If we have too much heat, we can see that the structure of the protein starts to break down. Imagine when you cook an egg, the whites of the egg are made of protein. As you cook that egg, the whites change from being colorless or translucent to being white. And that is because the protein is denaturing. Enzymes are made of protein, and so they denature as well. Now, it's really important that we say that the enzyme is denatured here. We're not going to get any marks if we say that the enzyme has died or that the enzyme has been killed, because enzymes aren't living things. They are just molecular machines, we could imagine. And when we say that it is denatured, specifically what happens is the active site changes shape. And if our active site changes shape, that means that it is no longer going to be complementary to the substrate. And if it is no longer complementary to the substrate, the enzyme substrate complex can no longer form, and so the reaction stops. And we can see that in our graph, where if we have temperature on our x-axis and our rate on our y-axis, in our first part of our graph here, part A, increasing temperature increases the kinetic energy of the particles, which means there are more collisions, and so more enzyme substrate complexes form and the rate increases. At point X, we have the optimum of the optimum temperature, where the rate of reaction is at its maximum. And then in part B, we can see that the rate starts to decrease. And where the rate is starting to decrease, this is because the enzyme has denatured. Our final factor affecting the rates of enzymes is pH. Now, pH is a measure of the acidity or alkalinity of a solution. And enzymes are adapted to function within a range of pH values. So, for example, our amylase enzymes, they are produced within the salivary gland, the pancreas, and the small intestine. So they are adapted to function within a ever so slightly alkaline pH, around about 7.5. Our protease, however, proteases, they are produced within the stomach. And the ones that are produced in the stomach are within a highly acidic environment, a very low pH. And so they are adapted to function in that acidic environment. So what happens outside of the normal pH range? Well, the enzyme is going to denature again. And we can see that in our graph here, where as the pH changes along our x-axis and our rate on our y-axis, outside of this optimum range, so at extreme pH, the enzyme denatures, and so the rate of the reaction decreases. We can see our blue line here, we've got a slightly kind of smoother curve or a slightly wider curve. And that's because this enzyme is going to be adapted to work at different pHs. And that might be for an organism that lives within a very changeable environment. So our different factors that affect the enzyme, these are super important, and they're going to come up time and time again in different exam questions. So you need to always keep these in the back of your mind. If you see a question asking about temperature, you need to be thinking, well, that's going to be affecting enzymes. If you see a question about pH, you need to be thinking that's going to be affecting enzymes as well. In addition to our enzymes, different parts of the digestive system produce other substances that help to aid in digestion. One example of that is bile. Now, it's really important to remember that bile is not an enzyme. Lots of students make that mistake. Bile is produced in the liver and then stored and secreted from the gallbladder. And it is secreted into the small intestine. Now, when it is secreted into the small intestine, that is going to be just after the stomach. Now, the stomach, remember, contains stomach acid, which is a very low pH. And if the enzymes in the small intestine were exposed to that stomach acid, or perhaps the enzymes from the pancreas, they would denature. So one of the first roles of bile is to neutralize the stomach acid. And to neutralize that stomach acid, it is going to then provide an optimum pH for the enzymes in the small intestine or the enzymes from the pancreas. So it provides an optimum pH. So the bile also has an additional role, and this is specific to the digestion of lipids. Now, the bile also acts to emulsify those lipids. Now, the process of emulsification is when the large lipid droplets, which are insoluble, are broken down into smaller droplets. Now, these smaller droplets, they are going to have a much larger surface area. And like when we think about chemistry and rates of reaction in chemistry, if the surface area increases, that means that there is going to be a greater rate of reaction. So this enables the rate of digestion of lipids to increase. So increasing the digestion of lipids. So we mentioned that the digestive system has two main functions. Digestion, breaking down the large complex molecules into smaller soluble molecules, but also absorption. Now, absorption primarily takes place within the small intestine, and the small intestine has some adaptations to increase that rate of absorption and to maximize absorption. Our first adaptation is our large structure here. This is called a villi, or villus for singular. The villi, coupled with the microvilli, which are the folds of the cell membrane, this is going to increase the surface area. And if we increase the surface area, that is going to increase the rate of diffusion and osmosis and active transport as well. And if you want more detail about that, go check out my other video where I break down everything in module 1. Now, alongside the microvilli, we also have a dense capillary network. So X here is our capillaries. Now, having lots of capillaries or having a good blood supply means that as soon as those soluble molecules like glucose or amino acids are absorbed, they are then going to be removed from the small intestine and transported elsewhere around the body. This means that the blood flowing through the small intestine is always going to have a low concentration of those soluble molecules. And so having this rich blood supply helps to maintain the concentration gradient. This is a point that lots of students forget to mention when they are discussing the adaptations of the small intestine. The fact that the rich blood supply helps to maintain the concentration gradient. Another adaptation of our villi or our small intestine is this exchange surface itself. The exchange surface we can see here is just 1 cell thick. And by being 1 cell thick, this is going to reduce the distance. And if we decrease the diffusion distance, we increase the rate of diffusion. Now, it's really important that we say that the exchange surface is 1 cell thick. Don't start saying that it has a thin membrane, because if you said that it has a thin membrane, maybe the examiner thinks that you think that it has a thin cell membrane, and that would be incorrect because the cell membrane basically stays the same thickness. Our final adaptation here is the mitochondria. And we can see that there are lots of mitochondria within these epithelial cells, the cells of this exchange surface. What is the role of the mitochondria? Well, they are there as the site of aerobic respiration. If you can't remember the different parts of the cell, again, go check out my other video where I break down everything you need to know for module 1. So the mitochondria are the site of aerobic respiration. Why is aerobic respiration important? Well, it's going to release energy, and it's going to release energy for active transport. So lots of mitochondria means that they can provide energy for active transport, and that is going to maximize the absorption of things like glucose or amino acids against the concentration gradient from the small intestine into the blood. So we've gone through and looked at the roles and the functions of all of the different parts of the digestive system. Let's now talk about experimental techniques we could use to investigate the digestive system and the function of enzymes as well. The third required practical in GCSE biology are the food tests, sometimes referred to as biochemical tests. And the purpose of these biochemical tests is to identify whether different biological molecules are present within a mixture or within a substance. We're going to be trying to identify starches, sugars, proteins, and lipids. Now, it's quite rare that you get a question that's just about the food tests. Quite often they will be integrated into other topics. So for example, we might use a test for starch to help us to investigate photosynthesis, because the plant is producing glucose in photosynthesis, which is then converted to starch. And that could help us to identify which parts of the plant are photosynthesizing. If you want to learn more about that practical or photosynthesis, go and check out my other video that I've made breaking down all of module 4. Okay, let's talk about our starch test first. Well, our starch test, we are going to use the reagent iodine. Now, the word reagent here just means the chemical that we are going to use to test with. Now, the iodine starts off as a orangey brown color. And if there is no starch present, it won't change color. It will remain an orange-brown color. If there is starch present, then it will change color to a blue/black color. It's really important that we include the black part there. So if you just said that it changes to a black color, you would still get the mark, but you'd have to say blue/black. You couldn't just say blue. You would not get the mark for that. Our next food test is to test for sugar. Now in this one, we are going to use our Benedict's reagent. Now the Benedict's reagent starts as this blue solution. And the Benedict's test has some additional detail associated with it. The Benedict's test, we are going to have to heat this. In fact, we're going to have to heat it to a pretty high temperature. It's best to say that we boil this solution, because that way we're making sure that we are going to get the mark no matter what the temperature required is. And we're going to do that using a water bath. And the Benedict's test here produces a range of different colours based on how much sugar is present. It will produce a green/yellow colour if there's a very small amount of sugar, transitioning all the way through yellow to orange red to eventually brick red if there is a large amount of sugar present. So we could say that our positive result, we have our brick red colour. Now protein, our next food test, is done using a Biuret reagent. And the Biuret reagent again starts as blue. Now this test is really easy to do. We just add some of the Biuret reagent and give it a shake. And if the protein is present, it will change to a purple colour. Sometimes we could say lilac as well. Our final test is our lipid test. And for the lipid test, there are two possibilities. Quite often people talk about the Sudan 3 test. Now, I'm not going to go through the Sudan 3 test because that's the additional one. The one you need to know is our ethanol test. So the reagent we are going to use is ethanol, and then we are going to also add water. The reason that I talk about the ethanol test is because you could be given a question that discusses using the ethanol reagent, and you would be expected to know what nutrient is being tested for. The Sudan 3 test, well, you could answer and say that the Sudan 3 test is used to test for lipids, but you're not expected to know the Sudan 3 test. Now, the negative result for our lipids would be that the ethanol just remains clear. Clear/colourless. If the lipids are present, the ethanol, when mixed with the water, is going to form an emulsion. We're going to end up with this cloudy emulsion being produced. So a cloudy white emulsion. This is why sometimes this test is referred to as the emulsion test. Our fourth required practical in GCSE biology is our enzyme required practical. And here our aim is to investigate the effect of pH on the rate of reaction of amylase. So our rate of reaction is how fast the reaction is progressing. And there's a few ways we could do this. We could measure our rate as our volume of product over time. Or we could measure our rate as 1 over the total time of the reaction. So 1 over how long it takes for all of the reactant to be used up and to be broken down. We're going to use the enzyme amylase to break down starch at a range of pH values, using iodine solution as an indicator for the reaction occurring. And as we just saw in our food tests, well, the iodine, if the starch is present, the iodine is going to change to our blue-black colour. If the starch has all been broken down, then the iodine does not change colour. The amylase will digest the starch into sugar, breaking it down. So the levels of starch will decrease over time, and eventually there will be no starch left. And we can use the iodine to act as an indicator of when all of that starch has been broken down, because once all the starch has been broken down, the iodine will not change colour. We can see here our breakdown of our starch and the fact that the amylase breaks the starch down into these sugar molecules referred to as maltose, and the maltose will continue to be broken down into the individual glucose molecules as well. Now, any experiment, we want to think about 3 different variables. We want to think about our independent variable, our dependent variable, and our control variables. Now, the way that I remember these is with the acronym CHIMED. And CHIMED stands for change independent. So the independent variable is what I change in the experiment. Measure dependent. So the dependent variable is what I measure in the experiment. And then the control variable is what we control. It's what we keep the same. So our independent variable, in this investigation, our independent variable is the pH of the solution. And how do we change that? We're going to use something called a pH buffer. So a pH buffer is going to change the pH and it is going to keep it at a constant level. Fortunately, we don't need to know how pH buffers work because they're quite complicated, and you'll learn about that in A-level chemistry. Our next variable is the dependent variable, and this is what we measure. So our dependent variable here is going to be our rate of reaction. But how do we measure our rate of reaction? Well, we're not going to be measuring our volume of product. What we're going to be doing is measuring our time for our reaction. So we're going to use the iodine solution to indicate the time taken for the reaction to take place. And how does it indicate that? Through the color change. So it's going to be the time for the iodine to not change color, because that is our indication that the reaction has finished, because there is no starch left. And then we need to think about our control variables. Now, when you think about control variables, you need to think about anything else that will affect the rate of the reaction, or anything else that will affect the dependent variable. So what else could affect the rate of an enzyme-controlled reaction? Well, it could be the temperature. And how would we keep the temperature the same? Well, just like we did with the Benedict's test, we would use a water bath. Now, I could change this experiment round, and perhaps I could investigate how temperature affects the rate of an enzyme, in which case I would want to change the temperature and I would then keep the pH the same. Other control variables could be the substrate concentration or volume. We want to make sure we are adding the same amount of starch. We also want to have the same amount of enzyme as well. So our enzyme concentration or volume. And then finally, this links back to our temperature, but when we mix our starch and our enzyme, well, if they're different temperatures, we're not controlling that, are we? So we want to make sure that they equilibrate to the same temperature before we mix them. And that way we are keeping that temperature controlled more accurately. So let's go through a final rundown of how we would conduct this experiment. Well, I'm going to start by adding my iodine to a spotting tile. As we can see, the iodine is orange or brown to begin with. Then we're going to add the amylase and the pH buffer to a test tube, mix, and then add the starch solution and start the stopwatch. We've let both of these equilibrate to the same temperature first. We're going to let the time run, and after 10 seconds, I'm going to add a drop of solution to iodine in the spotting tile. And we can see that we've got a color change. We can see that it has changed to a blue-black color. And that tells me that after 10 seconds, starch is still present. It hasn't all been broken down. So I'm going to do that after another 10 seconds. And I'm going to repeat every 10 seconds until the iodine doesn't change color. And we can see in this investigation, we've got 1, 2, 3, 4, 5, 6 there until it doesn't change color. Number 5, there is still a color change, a very small color change, but still a color change. So because there are 6, it was 6 different trials that we did, it's going to be 60 seconds for that reaction to complete. So our rate would be equal to 1/60 at a pH of 7. We're then going to repeat this using different pHs. And we can see there at pH 5, it takes 80 seconds. At pH 9, it takes 90 seconds. At pH 14, we can see that every single one of them, there is a color change. And that might mean that the enzyme is not breaking down the starch. And why might the enzyme not be breaking down the starch? Well, at this extreme pH, perhaps the enzyme has denatured. And for that reason, there is no reaction. We also need to know how we can improve the quality of our results in this investigation. Let's start by talking about how we can improve the precision of our data. Now, our precision is all about how much spread there is in the data caused by random error. And in order to reduce the effect of that random error, I would need to take repeats. So I would repeat at each pH. I could then remove anomalies. And I can then calculate a mean. It's really important that when we answer questions asking about how to improve the precision of the results, we say all three of these things, even if it's only a 1-mark question. What about our accuracy? Now, our accuracy here is going to be how close the optimum pH is to the true value. Now, let's imagine that the optimum pH for this amylase enzyme is 7.3. Well, from our experiment, we've identified the optimum to be pH 7. But I don't know if it's between pH 7 or pH 9, or pH 7 and pH 5, because I haven't tested any of those intermediate values. So one of the best ways to improve accuracy is to use smaller intervals. So use smaller pH intervals. In addition to that, this color change here, this color change could be subjective. How do I know whether or not the iodine truly hasn't changed color? So perhaps using a quantitative way, a numerical way of measuring the pH, of measuring the change in the absorbent, change in the color of the iodine. So what we could do, another way we could improve the accuracy is by using a different way to investigate the presence of the product. So quantifiably measuring, I'm just going to stop talking about that because I can just cut it out. I don't need it in this instance. Okay. So there we go. All of module 2 broken down and explained. Give this video a like, put in the comments any other topics you want me to break down in more detail, and make sure you check out all of the other modules too.